Hi friends, so this video we are just going to discuss origin of new genes and evolution of multigene family. So how the new genes are formed? It is formed as a result of gene duplication or the variation. So for an example, here humans have around 400 gene coding for smell receptors. The gene encoding these polypeptides or the proteins are phylogenetically related and have diverged gradually as the duplicate gene acquire new gene function. This mode of evolution may be called as the divergent evolution. As we know that the evolution of the life started from the water. The paradigm of the evolution of multigene family before 1970 was that of hemoglobin, alpha, beta, gamma, delta chain of and myoglobin. So, multi-gene family comprises of gene in an organism which are identical or having similar sequence, while the individual gene of the same multi-gene family usually have different function. So, the gene often become duplicated during the evolution, and duplicate genes sometimes acquire different function. The classic example of gene duplication come from the study of the globin genes in an animal. So, we can see here the example of organization of globin gene families in several mammalian species like human, mouse, and rabbit. Here we can say that the two genes that underwent repeated duplication giving rise to the various alpha-like and the beta-like genes found in vertebrates today. Repeated gene duplication such as that giving rise to the globin gene family appears to be frequent evolutionary occurrence. Duplication may also arise through also through transposition, also known as transposon genes or jumping genes, creating a pseudogene. In other cases, the change of the nucleotide sequence may lead to different function for the protein product of a gene. About 300 to 350 Maya, the alpha and beta globin genes were separated from each other and took residence on different chromosome. We are taking an example of hemoglobin. In human, for example, 7 alpha globin genes clustered on together on chromosome 16 and 6 beta globin genes are clustered together on the chromosome 11. So chromosome 16 contains 7 alpha globin and chromosome 11 contains 6 beta globin gene. So 3 of the alpha and 1 of the beta gene in human are non-functional, that is pseudogenes, and the other hemoglobin gene in this cluster encode different but related polypeptide. They carry oxygen in the blood at different time during life. So another phenomenon that might help to explain phenotype evolution is that the portion of the gene may be duplicated and recombined with other genes. Walter Gilbert speculated that each exons in a gene, that is functional gene, encodes a separate functional domain in the gene's polypeptide product. We know that the functional genes are formed by the removing of the introns and adding of the exons, that is exon shuffling. Tissue's plasminogen activator, the TPA factor, hemoglobin factor number 3. So for an example, exon shuffling in TPA gene, tissue plasminogen activator factor. One exon come from the gene fibronectin, another from gene for epidermal growth factor 2, exon from gene plasminogen, and one from the gene Prurokinase. Altogether, 4 genes contributed exons to the formation of TPA genes. As a conclusion, we can say that in addition to gene duplication and exon shuffling, evolutionary diversification seems to have benefited from spatial temporal changes in the expression of the genes. Example of the multi-gene family, the most prominent example includes gene for the rRNA, that is ribosomal RNA in eukaryotes. In human gene alone has 2000 gene of for 5S RNA. They are 18S, 5.8S, 5S, and 28S rRNA. So 5S rRNA includes 2000 genes. Multigene family of exinus, immunoglobins, interferons, tubulins, hemoglobin, histones, etc. have been identified. Multigene can be classified into orthologs and the paralogs. We have already discussed about the orthologs and paralogs. Genes are diverged from these different species but maintain the same function is known as the orthologs, while paralogs are just opposite of that. More than two multigene family form the supergene family. For an example, hemoglobin. As a conclusion, we can say that the new genes have evolved as a result of gene duplication and divergence. Since the individual genes of a same species diverge faster, the sequence similarity between species is more compared to the similarity of the gene sequence within the species. The speciation and the divergence event can be calculated from the analyzing similarities, and this form the basis of the multigene evolution in a construction of the phylogenetic tree. This is all about the new genes and the multigene family of evolution. Thanks for watching.